This is Amanda Welch welcoming you to this Bite-Size Bio web seminar, which today is sponsored by GenScript. GenScript is the leading gene, peptide, protein, and antibody research partner for fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. Since their establishment in 2002, GenScript has exponentially grown to become a global leading biotech company that provides life sciences services and products to scientists in over 100 countries worldwide. During their tenure, they have built the best-in-class capacity and capability for biological research services, encompassing gene synthesis and molecular biology, peptide synthesis, custom antibodies, protein expression, antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Today's presentation is titled Understanding and Designing Flanking Homology DNA Assembly Experiments, and is being presented by Dr. Nathan Hilson, who is the Director of Synthetic Biology Informatics at the Joint Bioenergy Institute. Nathan received his PhD in biophysics from Harvard Medical School in 2004. He was then a postdoctoral fellow at the Stanford School of Medicine. And in 2009, he became the Director of Synthetic Biology Informatics at the Joint Bioenergy Institute. His research focus is on developing and demonstrating experimental wetware, software, and laboratory automation devices that facilitate, accelerate, and standardize the engineering of microbes. Now, as always, we'll have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Nathan at the end. The recording of the webinar will be available within the next 24 to 48 hours at bit.ly/DNA assembly, all lowercase, all one word. That's bit.ly/DNA assembly. So now over to you, Nathan, for the presentation.
All right, thanks a lot, Amanda. And thanks to David and GenScript for sponsoring this webinar. As Amanda mentioned, I'm going to be telling you guys a little bit about DNA assembly. I'm spending a little bit more time on flanking homology methods, and then kind of touching upon protocol design software and synthetic DNA. So just as kind of a brief outline of where we're going to be going, I'm going to be starting out with what is the actual challenge with DNA assembly and where DNA, synthetic DNA fits in. As a little bit of background and context, I'll spend a couple of slides on traditional and BioBrick approaches to DNA construction, then kind of getting into flanking homology methods, including Gibson assembly. And then for some additional context, also talking a little bit about some of the type 2S methods and the user approach. After we kind of get through some of these methodology types of introductions, talking a little bit about some considerations around the pieces that you would be assembling together and what to look out for if they're going to be incompatible with each other and maybe how to resolve some of those incompatibilities. And then kind of starting to conclude on the methodologies, just some takeaway messages from what's been presented so far. And in the end, I'll just give you a very kind of a quick flavor for kind of available software design tools that might help you design your own DNA assembly protocols. All right, so kind of at a high level, the DNA assembly challenge is shown on this slide. So at the bottom, we might have a variety of different types of DNA fragments that we might want to be putting together. So we might have something like a vector backbone, we might have components like coding sequences or promoters, terminators, and we basically just want to assemble all of these together into, say, for example, a circular plasmid construct or a linear cassette that maybe we would be integrating into a genome. So traditionally, the way that this has been done, and this is kind of what I did in graduate school, is that you would have an expression vector. So the PET vectors are kind of a popular series of vectors. And within these cloning vectors, you would have a multiple cloning site. So this might be a series of, say, 10 different restriction enzymes, which are kind of unique cutters within that enzyme. And if you wanted to clone in any particular insert, you just looked for a couple of restriction enzymes that are in the multiple cloning site, but absent from your particular insert of interest. And this works great for just single inserts. But if you want to start putting together multiple inserts at the same time, or if you want to start thinking about automating your cloning with robotics, it becomes really challenging because any particular cloning approach is going to be using different combinations of enzymes, so that becomes challenging to make a uniform master mix across all of your assemblies. And as well as you get increasing numbers of inserts, it becomes really hard to find additional restriction enzymes which are absent from all of your inserts. And there's some additional challenges we'll get into in a second in terms of scar sequences and things like that. So maybe kind of one step beyond the traditional approaches is the BioBrick approach. And what I'm showing in particular is actually the BagelBrick kind of variation of the BioBrick approach. Several publications kind of going back into 2008, 2010. And the basic idea here is that can we make an approach that's kind of much more standardized and automatable? And the idea here is that basically we have kind of these standards where flanking every DNA fragment that you might want to assemble together, you might have two upstream enzymes. In the BagelBrick approach, it's EcoR1 and BAGEL2. And then downstream, you have a BAMH1 and ZO1. So if you want to basically put together part A with part B, you can, for example, digest the part B vector with BAGEL2 and ZO1 to release that fragment. And then in the part A vector, you digest with BAMH1 and ZO1. And the really clever thing here is that the overhang sequences that are released from BAMH1 and BAGEL2 are the same. So they're basically kind of cohesive and sticky. But after you fuse that set of overhangs together, you now have a sequence that can no longer be cleaved either by BAMH1 or BAGEL2. So now kind of your product sequence, again, matches kind of the same packaging, where you still have a unique EcoR1 and BAGEL2 upstream and a unique BAMH1 and ZO1 downstream. Now, this is automatable and it's standardized, but again, kind of pay attention to this scar sequence, because every single time you fuse together two BioBricks, you're going to end up with one of these scars. And the other kind of limitation with BioBricks is you're only able to basically put together two things at a time. So if we have kind of a more complicated thing that we want to put together, so maybe we're starting with 1, 2, 3, 4, 5, 6, maybe 7 different parts that we want to put together, we have lots of kind of, just two at a time assemblies that we have to do. So you have this binary assembly tree with lots of steps. And also every single time you put together two parts, you end up with this scar sequence, which you can't control. So those might be some of the limitations of kind of the BioBrick approach. The other kind of maybe things just to think about is that it's easy to add things to the ends of BioBricks, but basically you're stuck with what you have in the middle. It's very difficult to go back and modify pieces in the middle unless you're going to go way back to the beginning. And if you have any combinatorial diversity, so say maybe we had lots of different variations of this ORF-D, so we had lots of orthologs, or we had different RBSs or different terminators. Since each one of these steps is essentially a cloning step, unless you collect lots and lots of colonies over and over again, you might lose a lot of your combinatorial diversity. So those are some of the considerations. Now, beyond BioBricks, there's a large variety of kind of more modern approaches that are kind of more sequence independent. So this is just one kind of flavor of these flanking homology methods, and this is slick. So they call it sequence and ligation independent cloning. So the idea would be that you might have some type of a linearized destination vector. You might derive this from PCR or a digest, for example. And then you have a PCR product, or it could even be actually a synthetic piece of DNA that you could just order directly from a company like GenScript. And the idea here is that kind of shown in white is homologous sequences between the destination vector and the PCR product, and then also in gray. And with Slick, essentially it has a 3' exoactivity of the T4 DNA polymerase in the absence of DNA TPs. So you start getting some chewback on the 3' ends. And then you basically add in DCTP to kind of arrest kind of the chewback activity. Now you have these kind of cohesive sticky ends where kind of this white overlap sequence is going to be able to stick together with the corresponding white overlap on the destination vector. And then basically these guys just kind of stick together, and then post-transformation inside of the cells will repair these single-stranded DNA gaps. Now, if you look at something like the Gibson approach, it's very related. Same basic topology, so you basically start with exactly the same inputs. So you have a destination vector and a PCR product, again with the kind of the flanking homologous sequences. Gibson works slightly differently than SLIC. Instead of a 3' exo, you have a 5' exo activity. After a while of the exo activity, you reveal these single-stranded overlap sequences. And the one distinction between the Gibson approach and SLIC is that in addition to kind of the 5' exo, you also have say a fusion polymerase. So after you have this annealing event, the polymerase can basically start to seal all of those gaps. And essentially you have a race condition between the exonuclease, which is chewing away, and then the polymerase, which is trying to fill in. Now the polymerase is heat stable, and the 5' exonuclease is less stable. So over time, this activity for the exonuclease is going to go away, and the polymerase kind of maintains. And then you have kind of a Taq ligase at the end to kind of seal the nick, and then you do your transformation. So very similar type of an approach. There's other methods too, like this is the CPEC approach, circular polymerase extension cloning. Again, very similar types of input sequences. The way that this works is a little bit different. You actually, it's almost like a kind of PCR thermal cycling, where you kind of melt the double-stranded DNA into kind of single-stranded, and then maybe the top strand of this PCR product or synthetic DNA fragment would then be annealing with maybe the bottom strand of the vector. So it's kind of a different way of kind of getting these guys together, but you still have the polymerase, which will then basically go all the way around the plasmid, for example, and then you're just left with these couple of nicks, which gets fixed when you transform. Just as kind of another type of an approach. This is kind of yeast transformation associated recombination. I guess the figures that I grabbed here are from the Shao 2009 paper for DNA assembler, but the idea is very similar. You have these flanking homology sequences that you basically have guiding the assembly process, and this can kind of shown on the left side. This can, for example, be making circular constructs, maybe with a 2-micron plasmid for yeast. Or you can also imagine having a DNA fragment that's assembling together, but doesn't completely make the full circle, but could then be crossing over, for example, with the genome. So you might be able to use this to integrate entire multi kind of fragment pieces of DNA into the genome. So by no means an exhaustive list, but those would be some flavors of the different types of assembly methods. And we'll talk about a little bit later why you might want to be using one rather than another in particular circumstances. So again, just to kind of emphasize, there's lots of similarities between these methods. They generally all start with exactly the same types of starting materials, and they all basically end up with the same assembled products. There might be some slight differences in terms of how long these overlap sequences are going to be. So maybe if you have a method like SLIC or Takara CloneTex and Fusion, they're going to be a little bit shorter. Maybe Gibson might be kind of an intermediate length, and then maybe for the yeast tar cloning, you might have even hundreds of base pairs of homology. So it kind of depends on the method. But the nice thing about all of these methods is it's very easy to automate them. So you always use exactly the same master mix if you're doing kind of the in vitro methods like Gibson. Or if you're using yeast tar cloning, there really aren't any reagents other than the yeast themselves. Another really key point that I didn't emphasize yet is that all of these methods are scarless. So you get exactly the DNA out that you design. So unlike maybe traditional cloning where you still have all the multiple cloning sites and you have the scars from actually doing the ligations, or BioBricks where you have all these scar sequences. If you look at these methods, like the flanking homology methods, you have control over every single base pair. And I think that's a really kind of a key value add of these methods. And it's also largely speaking, although we talk about some of the caveats there in a second, it's largely sequence independent. So you can basically use any sequences you want and assemble them together, including if you want to, for example, use methods like the flanking homology methods to build BioBrick compatible vectors. Or as we'll see later, you could use a Gibson approach to assemble together a Golden Gate type of a vector. So that's also kind of a really nice thing, is they're largely sequence independent. Now, kind of going back to the DNA assembly challenge, it might be that we don't only want to put together one fragment at a time. You might want to put together multiple fragments. And unlike BioBricks, where you're limited by putting together two fragments at a time, or with traditional cloning, where it becomes increasingly difficult to add in each additional fragment, some of these flanking homology methods, you can say, for example, get up to maybe 10 or so fragments going together at the same time. It's increasingly difficult. Once maybe you get beyond 4 or 5 fragments going together at a time, it does become a little bit more difficult, but you can get larger numbers of fragments going together. And the way that this works, you basically just follow the rainbow in this particular example. So like colors go with like, and you basically just follow the colors all the way through, and then it basically will assemble into the final construct. Other things to kind of highlight, it's just as easy to change, something in the middle of a pathway as it is things at the end, which is kind of differentiating from BioBricks. And if you want to do kind of combinatorial libraries, since you're doing this all in one step instead of multiple cloning steps, you can basically not lose any of your sequence diversity between steps. So you still have to collect, as many colonies as you're going to need to oversample the diversity of your library, but you're only doing a single cloning step, so it's easier to not have to worry about losing your diversity. Now, some of the challenges here would be that you really do have to design all of the overlap sequences to make sure that, for example, that you only get like junctions assembling with like. And in order to basically add on all of these flanking homology sequences, you might have to do that, for example, through DNA oligos and PCR, or just ordering a synthetic DNA fragment that already has these flanking sequences. So there is, some work that that needs to be done, and you also have to be careful, as we'll see in a second, about having repeated sequences. So, say for example, and we'll see this in a second again, if you have two, PLAC promoters, how do I differentiate this yellow sequence from this bluish sequence? So there's definitely some challenges still. As some additional kind of context, and maybe kind of reflect on like why the flanking homology methods are advantageous in certain situations, it's probably useful to also talk about some of the type 2S methodologies. This is the Golden Gate approach. You can kind of see some of the Engler papers that kind of first reported this. And the general concept with these type 2S enzymes is that the recognition sequence shown here is BSA1. The recognition sequence is separated from the actual overhang that gets generated. So you basically have full control over any overhang that you want to use. So the way that this is kind of done in practice is that you can, for example, start with the destination vector that already has kind of these BSA1 sites in the vector, or you could prepare it through PCR linearization. And the same thing with the PCR product. So instead of maybe having a flanking homology sequence at the ends for something like Gibson, you have kind of these flanking BSA1 sites, and then your different types of overhang sequences. And besides PCR products, this could be a synthetic DNA fragment that you potentially just order. Now, one way of implementing kind of the Golden Gate approach, and this was probably the first way that it was reported, is that you can put all of these fragments together in a single pot with BSA1 and ligase, and essentially you kind of have an alternating type of a thermocycle happening, where at kind of the optimal temperature for BSA1, you're going to be trying to digest, so you're going to be making cuts at all of these overhang locations. And then you have a ligation step where you can basically take the cut parts and ligate them together. Now, not only is it possible to kind of go to your final product, but you can just kind of reversibly ligate back to get to your original starting material. However, for the final product, there aren't any BSA1 sites left in the final construct anymore, so you can't digest this. So this is effectively a dead end product. So over time, after several thermocycles, your population of molecules will kind of evolve towards this desired end product. Now, a couple of things just to point out. So this isn't quite as sequence independent as maybe some of the flanking homology methods, although we'll caveat that in a second. But essentially, you don't want to have, ideally, you don't want to have the same BSA1 site anywhere else in either your kind of the insert or into the vector, because if that's the case, then basically every single time you're going to be chopping apart your insert or chopping apart your vector. Now that's not that's not going to defeat the process, because as long as you end on a ligation step, you can still end up with your final product. But it just makes the overall process a little bit less efficient if you have internal BSA1 sites. Now, just as kind of with the flanking homology methods with Golden Gate assembly, you can be putting together multiple fragments at the same time. Again, just kind of following the colors as you kind of go through the process. Some of the advantages I would say of kind of the Golden Gate assembly approach is that these overhang sequences are extremely short, so only say 4 base pairs. So if you want to get full combinatorial reuse across an entire library, so you only want to maybe prepare a single PCR product of the red insert once, or you only want to have to order a synthetic fragment containing that red fragment once, then with Golden Gate, the only requirement is that every different variant of your library has essentially these 4 base pairs of identity across all the combinations. Whereas, for example, if you're trying to do that maybe with the Gibson approach, this might need to be closer to say 25 or 30 base pairs. So if you're doing protein fusions or types of things like that, it might be advantageous. Just as kind of like with the flanking homology methods, you do have to kind of make sure that all of these overhangs are going to be compatible with each other, and we'll talk about that in a second. You still have to design kind of all of this flanking sequence and the oligos for making those. You have to design the synthetic DNA fragments for doing those. And then it's also kind of really important to kind of find the most cost-effective design strategy. So depending on specifically where you place these overhang sequences, you might be able to get full combinatorial reuse. Ideally, if you're putting it in a region that there's lots of sequence identity, or if there's no sequence identity, then you're not going to be able to have as much reuse. So there's a lot of kind of fine tuning and optimization that you might need to be able to do to kind of get the best possible outcome. And again, just as kind of as a background context, in which we can kind of frame a discussion around the flanking homology methods. This is just kind of another type of related approach, the user approach. And here you essentially do need to do some type of a PCR reaction or do DNA synthesis that's enabled by having uracil base pairs. And there's a particular set of enzymes, I guess they're kind of collectively referred to in this method is just the user enzymes. And the idea is that the user enzymes will cleave at a uracil position, which basically generates these overlaps. And the kind of cool thing about the user approach is that you can control exactly how long this overlap sequence is. So for methods like the type 2S methods, generally you're going to be limited to whatever the enzyme is going to be cleaving. So it's typically say 3 or 4 base pairs. And then user can be, say, anywhere between, say, 8 and 15 base pairs, which is going to be a slightly shorter length than you would find with the flanking homology methods. So it might be able to provide a little bit more specificity than, say, a Golden Gate or type 2S approach. But it's going to be shorter than maybe a flanking homology approach. So if you have kind of shorter regions of sequence identity, maybe this might be kind of a nice sweet spot in between a type 2S approach and a Gibson approach. Now, I think what we need to talk about a little bit are some of the challenges in some of the incompatibilities that you might encounter between the assembly pieces that you're putting together. So this is a specific example, and we'll kind of maybe talk about it from the aspect of maybe a Gibson assembly or other type of flanking homology method. But let's say that we're trying to put together this particular construct where we have maybe a vector backbone, and then we have kind of 6 inserts that we want to put together. And as you kind of see here, maybe the insert number 2 and the insert number 5 are the same PLAC promoter. So a challenge is if I'm kind of assembly piece number 1 and I'm looking downstream for something that looks like this PLAC, you know, I could potentially just assemble together with insert number 2, or I might mistakenly instead assemble together with insert number 5, because basically 2 and 5 look very similar together to each other. So instead of basically having the full vector going through inserts 1, 2, 3, 4, 5, 6, I might basically have the vector and then insert 1, skip all the way to insert 5 and 6. So I'd be missing 2, 3, and 4. So that wouldn't be what I want. So you could potentially get these side products that you don't necessarily want. So one way that people can get around some of these incompatibility issues, for example, is doing what we call a hierarchical assembly. So you can imagine that we could first, say for example, do a splicing by overlap extension or PCR fuse fragments 1 through 3, and then separately 4, 5, and 6. And then basically we would just be assembling these 3 pieces together. And that way that kind of the ends of these contigs, whether it's the kind of the 5' end of assembly, piece 1, or the 3' end of assembly piece 3, there aren't any incompatibilities at these ends. And basically what we've done is inside of these contigs, we've kind of buried all of these junctions, which basically are repeated somewhere else in the construct. So this might be a way of actually trying to get away from some of these assembly piece incompatibilities. But there's actually quite a bit of work that might be required to actually figure out when these ends are compatible or not. So for example, one thing that you might do is just look at all of your flanking homology at the ends of each of these assembly pieces and then just blasting them against all of the other assembly pieces. And for example, it looks like in this case, if I blasted the kind of the right side, the 3' end of assembly piece 1 against all of the other assembly pieces, I might have had some homology to inside of assembly piece 0. And that's because there's also a PLAC in the vector backbone. And then kind of the 4 or 5 junction also has this PLAC sequence. So I would find out that I probably don't want to put in the right side of assembly piece 1 in any assembly reaction that contains pieces 0, 4, or 5. So basically what we did is we first assembled together 1, 2, and 3. So now the right side of number 1 is no longer exposed. And that's basically how it works. So it can be time consuming and tedious. But if you really want to make sure you avoid any of these incompatibilities, you kind of need to do those steps. Now kind of shown kind of here is more of a Golden Gate type of an example, but it's exactly the same process that you would use for flanking homology methods like Gibson or others, or even user cloning for that matter. So the idea is you really want to make sure that your overlaps or overhangs are matching between parts that you want to go together. You want to make sure, for example, that the kind of the blue overhang sequence of the red piece is going to be going together with the blue piece on the vector backbone. But what you don't want to have happen is you don't want to have, for example, a blue overhang being complementary to a red overhang, because that wouldn't be a desired thing. And I guess in this particular example, we have a case where we have a kind of a blue overhang that is overly complementary, say 3 out of the 4 base pairs, with something that's kind of an off target. So these might be the types of things to look out for. So for Golden Gate, you can or maybe for user, you can kind of count up the number of mismatches, and you might have a threshold saying, I can tolerate up to 2 matches, but 3 or more is too much. For things like flanking homology methods, you might be looking at maybe melting temperatures of the matches between things to see if they're going to be stable under your reaction conditions. But it's a very similar type of an approach where you want to make sure that things that are going to be going together that are intentional, whereas things that shouldn't be aren't. Now, one of the beautiful things about these methods, whether it's Golden Gate, which is shown a little bit more here, or the flanking homology methods like Gibson, is that you have a lot of different choices in terms of where to place your overhang sequences for type 2S methods like Golden Gate, or your overlap sequences like Gibson. And at the end of the day, all of these different choices are still going to result in exactly the same assembled sequence. So you're still going to be completely scarless. You're still going to end up with exactly the same product that you want. But the way that you actually implement the design process or the assembly process is a little bit different. So this is for Golden Gate, but it's exactly the same idea for flanking homology. So if we have 2 parts that we're putting together, say the gray part and the white part, One thing that we could potentially do is choose a very neutral overhang location. So maybe we're grabbing the first 2 base pairs of that overhang from the preceding part, and we're choosing the next 2 base pairs of that overhang coming from the subsequent part. So it's very neutral. It's kind of placed right in the middle. So if we assembled these guys together, we would get this final product. However, we can basically shift that overhang to be entirely contained within the preceding part, and it would still result in exactly the same product. Or we can shift the overhang, down into the subsequent part, and again get the same thing. So basically, you can increasingly shift the overhang out to the left into the preceding part, and you can shift it out to the right into the next part, and you still get the same thing. So this is really key for several reasons. One of the reasons is that if you can shift around the overhang or overlap sequence and still get the same sequence, that basically allows you to make sure that the overhang or overlap are in regions where, for example, in a combinatorial assembly process, you can locate these overhangs or overlaps where there's lots of sequence identity, so you can really maximize the reuse across all of your combinations. And the other thing is, for example, if you saw that maybe with purely neutral overhangs or overlaps, if you do get some of these incompatibilities in between the overhangs that you've selected, all that you really need to do is start shifting the actual overhangs or overlaps until you look across all pairs of junctions, and everybody is only going together with who they should be going together with. Now, again, this can be a little bit tedious and time consuming, and if you don't have software that can help automate that for you, that might be pretty laborious. I should just kind of note in passing that I've been emphasizing a lot scarless approaches, where you always get exactly the same sequence that you want. And the trade-off here is it really does take a little bit more effort on the design side to make sure that all of your processes are going to work efficiently. Now, there are some methods that are kind of a little bit more standardized in terms of always using exactly the same overhang sequences or overlap sequences. So I guess in flanking homology methods, you might call those kind of standard linkers that you would use, or for type 2S methods, standard overhang. So if you look at methods on the type 2S side, like Moklo or other types of standard methods, they basically pre-validate all of the overhangs. So you don't have to do any thinking at all around if your overhangs are going to be compatible or not. That's basically already been solved for you. The really big challenge there, though, is that you're always going to end up with these scar sequences of these overhangs that you didn't have any control over. And it's the same thing with the flanking homology style methods, where if you use these standardized linkers, then again, you don't have to make sure that the linkers are compatible with each other. But then you have all of these scarred sequences, all these linkers that you don't have any control over. Right. So just some take-home messages, I would say, for the DNA assembly methods. And the first thing is just kind of a point about the role of DNA synthesis. So a lot of places I mentioned that you could be deriving these fragments from digest or from PCR reactions. It's very common practice now where you could basically get to that directly from DNA synthesis from vendors like GenScript. So that can really kind of facilitate your workflow. There's lots of different methods to choose from. I guess here we talked mostly about flanking homology methods, and say, for example, between those different methods, there might be different kind of advantages. So maybe the different methods have different overlap lengths. So maybe infusion might be a little bit shorter. Maybe Gibson is more intermediate. If you have something like yeast assembly, maybe very long. So the longer the sequences, you can potentially get more specificity at the trade-off of combinatorial reuse. There's some other types of considerations. For example, with kind of maybe like the CPAC method that we talked about before, if you have the entire DNA strands separating and then re-annealing, potentially you could get misannealing anywhere along the DNA assembly fragments, whereas with some of the more, I guess we refer to them as two-back methods like Gibson, you really only have to worry about maybe some mispriming or misannealing at the very ends of your pieces. In terms of kind of sequence independence, and we talked a little bit about how Golden Gate is not as sequence independent because you have to make sure that you don't have restriction sites. However, for some of these flanking homology methods, you still have to worry about things like DNA hairpins. So you are revealing, for example, in Gibson, single-stranded DNA at the ends of your pieces. And if you have really strong hairpins, basically that single-strand DNA might just anneal to itself as opposed to going to its partner. So you have to think about things like that. Now, secondary structure might have different temperature, melting temperatures. So if you have methods maybe like infusion or slick that are operating at kind of maybe lower temperatures, maybe Gibson is slightly higher temperature and then CPEC would be the highest temperature. Depending on the temperature of your assembly, you might be able to get rid of some of these secondary structure issues. I guess, which is what kind of mentioned right here. I think kind of going forward in vivo assembly, like yeast TAR cloning, is going to be really important because generally speaking, that's going to enable you to get to much larger pieces of DNA and potentially more pieces of DNA because of the specificity and an organism like yeast capacity to propagate larger pieces of DNA. I think there's a really big philosophical divide between kind of standardized design patterns, like we talked about, BioBricks. We kind of reference in passing things like standard linkers for things like Gibson, or the MOCLA methods for things like Golden Gate. And there's some advantages here. It kind of facilitates the design process, pre-validates overhangs or overlaps against each other. But then you're kind of stuck with all of these scar sequences, and you're kind of trapped into a given paradigm. So it's kind of flexibility and making exactly what you want versus facilitating some of the design work. All these modern methods, I would say, are fantastic in that they really do enable you to scale your automation processes. So it's easier to implement things on laboratory automation or even microfluidic devices. So that's been a really great thing. And algorithmic design optimization can be very beneficial. That can really help you, especially if you want to be flexible. That can really take off all of the burden from you from designing all of the protocols. It can make sure that you don't have to worry about incompatible assembly pieces and all these types of things. And I think coming up right next, we'll be talking about maybe some of the software tools that kind of help you design protocols and at the same time allow you to maintain all of that, that great flexibility. So in terms of some of the software around DNA assembly, back in 2011, we published the J5 paper. So you can check that out in ACS Synthetic Biology. And if you want to kind of learn more about that, it's also so there's some documentation at j5.jbay.org. And this is largely kind of an academic piece of software. But over time, I think as of now, there's over 2,000 people around the world that have registered to use J5. But there's also lots of really nice commercial tools that are out there. So maybe I'll just touch upon two really briefly. So the first set of tools are very specific to particular methods. So this is maybe a primer design tool for Takara CloneTech for their infusion. So they basically would be designing protocols that are very fine-tuned for their particular reagents, and it's really nice, I think, for the users to have access to these types of tools because it takes away all of the effort from designing the protocols. But it also will increase kind of the success rates of the users. So it's great for the users because they're more successful, and I think it's also great for vendors because their users and their customers are generally happier, and it's easier to use their reagents. There's also some other platforms which are a little bit more general across different types of approaches. And I'm just going to give you maybe like a couple of minute demo of the Tecelligent platform just to kind of highlight some of these things and maybe how synthetic DNA can basically be incorporated directly in these software tools. So I'm just going to go ahead and exit PowerPoint for a second. And I'm just in Chrome here at the Tecelligent.com website. I'm just going to sign in to my account to kind of go into their application. And I think what I'm going to do is I'm just going to open up a design that I already have. And this is a very simple kind of combinatorial design. So we might want to be doing here is we have like a vector that we like, but maybe we want to swap out a particular antibiotic marker for other markers. So for example, in the current vector backbone that we have, Maybe we have chloramphenicol resistance, but we kind of want to swap that out for other types of markers. So maybe some of our other plasmids that we have maybe the gentamicin resistance that we want, but maybe it doesn't exactly have the backbone that we want. So all we're really kind of doing here in this design, which is very simple, is kind of inserting these 4 different markers into this vector backbone replacing chloramphenicol. Now, one thing I just highlight in particular is that in software tools like the Celogens platform, you can often specify the assembly strategy for particular components. And in this particular case, maybe for the gentamicin cassette, maybe we want it to be synthesized by a company like GenScript. So we can specify that here. Or maybe for some of the other parts, we're not going to really force any assembly strategy, and we'll just allow the software to kind of find out the optimal strategy. So should I do this with synthetic DNA because it's going to be most cost effective? Should I do it with PCR? If it's a small part, should I be embedding it in a primer? There's lots of decisions to be made, and generally speaking, the software can really help you make those decisions. Once you have maybe set which parts you want to have synthesized, for example, maybe we can just kind of go to the DNA assembly methods here. Maybe the method that we're going to choose here is a flanking homology method, like Gibson. And you can kind of use default parameters, or maybe just to kind of show you. A lot of these software tools are fairly configurable, so you could kind of customize it, for example, for infusion or for Gibson or for yeast tar cloning. So maybe for Gibson, we like 26 base pairs of homology, but maybe for infusion it might be a little bit shorter, or for tar cloning it might be a bit bigger, or maybe for type 2S methods we might be choosing different types of type 2S enzymes and so on. And if you have a great deal from a vendor like GenScript, maybe you can update the kind of the default prices per base pair of DNA and things like that. Well, maybe let's just go ahead and run this protocol. So it's going to take a few seconds to run, but essentially what the software is doing is looking at each one of these different fragments. And I guess the first thing that it's going to be doing is figuring out kind of the the cost optimal way of putting this thing together. So we already know that we're going to get the gentamicin cassette synthesized, but maybe for the other parts, is it more cost effective to order oligos or to get DNA synthesis? As DNA synthesis prices become increasingly competitive, the skew is going to be increasingly towards just getting it synthesized rather than doing PCRs yourself. And then after kind of it decides on the general strategy, then it might be designing all of the synthetic fragments for you, the DNA oligos, worrying about are there any compatibilities between pieces and so on. So that's basically what the software is doing. So now that the software is done running, we can kind of go to that assembly report. So we have pretty much 4 constructs are getting made, the vector backbones and the 4 different markers. We might just want to preview what those things look like. So here we can kind of see a preview of the first construct, which has the gentamicin resistant marker in here. That's basically what we wanted. We kind of scroll down through this thing. We can find like the synthetic DNA fragment that we wanted to order, and this is for the gentamicin cassette. So we kind of know how long it is, and we know what the sequence is. So if you just want to put it into a vendor ordering tool, like for GenScript, you can just take the sequence, and it's already designed for you with all of the flanking homology sequences already included. For any PCR reactions or for reamplifying DNA fragments that have been synthesized, the tools will, for example, design all the oligos that you're going to need to, again, order from vendors. The PCR reactions that you would be doing, eventually the assembly pieces that you're going to have on hand with all of their overlap sequences. And then finally, it'll kind of just give you some guidance as to, to make each one of these constructs, which are the different assembly pieces that you mix and match together. It'll also give you any particular warning messages that maybe it found when you're trying to do the assembly. So for example, you might be very interested in knowing if there's any repeats within the sequence that you actually are building, because maybe for in vivo stability issues, those would be very useful things to know about. If there's any primer mispriming that you might want to worry about, et cetera. So I think these tools can be very helpful in trying to automate the process for you, still enable all the flexibility, really take advantage of synthetic DNA from vendors, and so on. So that's just a kind of a quick taste of some representative software tools. So maybe getting back to PowerPoint real quick, just kind of in summary, just talking about the role of DNA synthesis, and this is going to be playing an increasing role going forward as it becomes very cost effective, and the lengths of the synthetic fragments become bigger and bigger. So I think we're really going to see a real conversion increasingly over to DNA synthesis. There's lots and lots of DNA assembly methods to choose from, and new ones are getting created all the time. There's lots of methods that I didn't get a chance to talk upon, like the BASIC method, or Amyris's kind of LCA type of an approach. And I also really highlight that having software that can help design these protocols for you can be extremely beneficial, not only to reduce time and labor, but to save money, and also to avoid any mistakes that might cost you months. So just some quick acknowledgements. I'd really like to thank kind of Bite Size Bio and Amanda for allowing this topic to be presented. David Lin and GenScript for sponsoring this talk. The funding for the J5 development and a lot of my support at JBay comes from the Department of Energy and the Office of Science and the BER office. And then just some of the software tools that I kind of touched upon is kind of the primer design tool from CloneTech and Takara, and the software that I kind of demoed was Tecelligent. So now I think what we're going to do is switch over to David to say a few words about GenScript.
Hi, thanks Nathan for that great presentation. I think we all learned a lot about DNA assembly and the variety of methods and softwares we can use to achieve our experimental goals. I just wanted to give a quick background on GenScript and how we can help in that process. As some of you may know, we're a leading biology CRO, as Amanda stated in the beginning of the presentation. And one thing we do like to focus on is our state-of-the-art production facilities and also our highly technically trained staff, with over 40% of our staff being scientists with PhDs or master's degrees. And so what this means for you is that during your ordering process and the production of your service, we always have a scientist watching over the process itself. And so that way you always get a highly technical evaluation of your order. On top of that, we've been around since 2002, and we've been in over 11,000 peer-reviewed papers that cite both our services and our products. So we've been peer tested and proven. And also we're a global operation. We not only operate in the North America region, we also ship over to Canada, or the Asia region, and also in Australia and the UK and all over the world, basically. A quick highlight on how we can help with your assembly process. We recently launched a new service for gene fragments back in May called GenParts. And what this service is pretty much, it's a precursor to our DNA synthesis, our complete DNA synthesis service, where what we deliver to you is a gene fragment in a PCR format. And what this allows for you is some flexibility and versatility in how you use those gene fragments. You can either use them independently, or you can use them as Nathan stated, in a assembly method to create full-length genes and pathways. And our fragments, if designed based off your standard and specifications, can fit any major cloning assembly protocol. They're fast and reliable, being that when we give you a stated deadline, we over 95% of the time do achieve that deadline. We have no sequence restrictions when accepting sequences, and we, as stated before, we have outstanding service in our PhD level customer support. I just want to let everybody know, come try our service today. We currently are running a promotion of 30% off all of our GenParts orders, and you can find out more online at our website, stated below at www.genscript.com /genparts-gene-fragments.html. Thanks again to Nathan and Amanda for hosting this webinar, and Nathan for presenting a very informative webinar on DNA assembly. For more information about our products, please feel free to shoot me an email at david.lin@genscript.com or visit us online at www.genscript.com. Thanks again, and I'm going to pass this back to Nathan and Amanda for some Q&A.
Thanks, Nathan and David, for a great presentation. We have a few questions from the audience, and if anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So right now we have a question from George, and he asks, how would you, and this is a question for Nathan, and he asks, how would you recommend purifying PCR products if throughput is essential?
Yeah, that's a good question. So there's a variety of different ways of doing that, and there's different trade-offs. So you might be able to do kind of very simple old school kind of ways, just using salt concentrations and glycerol or PEG and centrifugation types of ways of just kind of separating away kind of maybe the contaminating oligos from your PCR products. You could just use simple PCR reaction cleanup, types of columns, which can be great if you have very pure PCR reactions without any contaminating bands. At JBay, we've been using gel electrophoresis, and specifically kind of the Coastal Genomics / Hamilton Nimbus system for kind of automating some of the gel purification processes. If throughput is really essential, again, there's different trade-offs with each of these different categories. So maybe the gel electrophoresis is slower and more expensive. Maybe columns, generally are going to work, really great. You can do a multi-well plate format. But if you have contaminating bands, that might be problematic. And you might be able to just get by with very, old school approaches too. But again, there's going to be different types of trade-offs. But generally speaking, for our production processes at JBay, we've been using the Nimbus because we want to kind of have a universal type of a method that works for everything. So we've been generally going with that type of an approach. But depending on your needs, you might have different slides of trade-offs.
Okay, that'll make sense. And then next we have a question from Roman. And so he has a open reading frame with a 20 nucleotide randomized region. Do you know of any way to clone and insert right next to that randomized region?
So I guess the question that I guess I would have is if that randomized region is kind of within the insert, and if it's kind of within the insert, I guess the question would be if you already have, for example, a pool of fragments that have all of that diversity in it, and you could just kind of amplify that pool with the primers to introduce the flanking homology or Golden Gate sites and things like that. So that would be an approach. If you actually have that diversity at the very ends of your insert, or I guess you could even split up that insert into multiple fragments so that the sequence diversity is actually at kind of one of the junctions of your assembly pieces, then potentially you could encode that sequence diversity in the oligo itself. So when you're amplifying an insert or template that you already have, you could be not only introducing the flanking homology or the Golden Gate, type of adapters, but also all the diversity at the same time. So there's a variety of different things that you could do there. Maybe with companies like GenScript, you could actually just order the fragments with the flanking homology and all of the sequence diversity in there too. So I think there's a variety of things you could do.
Yeah, that sounds like he has a lot of options. And next we have a question from, and I'm sorry if I mispronounced your name, I think it's Chen Fu. And they're asking about the chemical reason for why you need about 15 to 25 nucleotides for homology for Gibson assembly.
Yeah, so largely it's going to come down to the melting temperatures. So if you look at a method like Gibson in particular, the operating temperature of that reaction is at 50 degrees. So in order to have kind of those sticky ends kind of be cohesive and stable at 50 degrees, they have to kind of be above a certain length. And really, it's not about the length, it's more about the melting temperature. So if you have very AT-rich regions, you're probably going to need to have slightly longer overlaps. But it's largely about the operating temperature. So maybe for infusion, the operating temperature is lower, so you don't need quite as long overlaps. Gibson's a little bit higher at 50. Maybe CPEC is operating more in typical PCR conditions, 60 plus. So you might need even longer overlaps for something like CPEC. So those would be some of the reasons. It's mostly a thermodynamic melting temperature type of an argument.
Okay, that makes sense. And then Isaac has a question about, this is just about your general thoughts. So do you have any thoughts about E. coli in vivo assembly?
Yeah, so there are methods like Slice And you can, I guess you would almost call that like a ex vivo type of an approach, where you basically have E. coli that's overexpressing maybe some like the lambda red types of recombinases or other types of recombinases. And you can basically just make a lysate and use that as your reagent instead of having to buy the reagent from somewhere else. Or you could potentially do the same with E. coli just in vivo. If you had basically the same background strain as people use for making the lysate, you could do that. The challenge, I think, with E. coli or smaller organisms, bacteria, is they just don't have very much surface area. So it's really challenging to do a transformation and have multiple fragments go into the same cell. Whereas with yeast, they're much bigger. So it's much more likely that you're going to get multiple fragments to go into the same cell. So that's generally, I think, why methods like SLICE use lysate as opposed to just trying to transform in the separate fragments separately into E. coli, because just the odds of getting two or more things into the same cell at the same time is pretty low with something like E. coli.
Yeah, it makes sense. It's more of about a size restriction as opposed to whether or not it can actually do it. And then we have a final question from Tammy. And so she's asking about the Teslagen app.
Yeah, so basically anybody can sign up for Teslagen. Generally speaking, it's going to be, I mean, you can contact the Teslagen folks. I'm sure Mike Farrow there would be more than happy to talk with anybody. But generally speaking, it's going to be available at no cost to academics or nonprofits or government. And then for companies, there's probably several different options. You can kind of use their application, like the one that I showed, or there can be kind of more custom engagement. So some companies are very secretive with their data, and as opposed to kind of like a cloud-based application, they really kind of would prefer more of kind of like a local deployment. So those are all kinds of possibilities. The Teslagen app right now largely supports any of the flanking homology types of methods. It can support things like the type 2S methods like Golden Gate, combinatorial things, all those types of things. There's some really interesting things in the work there. They're increasingly going to be supporting hierarchical design. What Teslagen's app probably won't do for you right now are some of the methods like the user method, or I kind of mentioned very quickly in passing, maybe methods like Amyris's LCA method, where you use an oligo to kind of bridge two different kind of blunt inserts. But if you're interested in those methods, you can always contact the Teslagen folks, and if you're one of their industrial customers and you're able to support those types of developments, I'm sure they'd be interested in that too.
Okay, and then she also wanted to know, she said like commercial vector backbone sequences presumably can be uploaded into the system. What about if you have plasmids or constructs that you've designed yourself? Could those be uploaded into the Teslagen?
Yeah, exactly. So that's what in the demo that I showed, I had just uploaded all of those plasmid templates. So you can upload anything that you have yourself, and you might be able to access some really nice commercially available vectors too.
That's great. Oh, and we do have one more question. This is from Donatus, and they ask about commercial assembly kits. So there's a whole bunch of them out there. Like there's Golden Gate and Gibson and all these commercial kits. Do you have any thoughts on them, or and do you prefer one or the other or anything like that?
Yeah, so there's some things to think about. I think to a large extent, here we've been using, for the flanking homology style methods in terms of commercial kits, we've been using some of the Gibson assembly mixes, either from SGI or from NEB. And then we've also been using Infusion. And I think some of the really nice things that some of the companies are doing now is that they have very high concentration formats for their reagents, and especially if you're using things like the lab-side echo, so some of these acoustic printers, having really high concentration reagent kits is really great, because then you can use more of the volume on your DNA assembly pieces as opposed to just the master mix. So that might be a decision for you too. But I think it's just the right tool for the right job, and maybe in your particular workflow or for your types of needs, one particular kit is going to work better for you. So I think it's great that we have lots of choices.
And I do want to add that it looks like GenScript is going to be launching such a product at the beginning of the new year. So you might want to check back with them, or if you're interested in the commercial assembly kits, you can shoot David an email. I think I did send that out to everybody. So it's david.lin@genscript.com. And so you can shoot him an email and he'd be happy to answer any questions about that particular kit that you might have. Okay. Well, that looks like the very last one of our questions. So that brings us to the end of the seminar. And so I want to thank you again, Nathan, for a fantastic presentation and a really great discussion. And I also want to thank our sponsor, GenScript. And finally, thanks to you, the audience, for taking the time to attend and listen in. If you've enjoyed the seminar and would like to view the video recording of this session, please visit the webinars page on bitesizebio.com. It should be available within the next 24 hours or so. While you're there, you can also see the other webinars that we have lined up for you on Bitesize Bio. So until next time, good luck in your research and goodbye from all of us at GenScript and Bitesize Bio.